I like to start with prokaryotic cell division because they're really simple and really easy, and that will kind of get us primed and prepped to talk about eukaryotes, which of course are less easy to talk about. So what we're going to talk about here is the process of binary fission that occurs in prokaryotes, and we're going to talk about how these special proteins um, allow this separation of these cells and how we see them in both prokaryotes and eukaryotes. Okay, so prokaryotes' genome is really simple. They have one circular chromosome, and this is what is held in that nucleoid region. It's not a nucleus because it's not membrane bound like we see in eukaryotes. Prokaryotes also have other sources of genetic material than the chromosomes. We call these the plasmids. These are very small, circular uh, molecules of DNA, and the prokaryotes can swap these between individuals in order to create some genetic variation because prokaryotes cannot do sexual reproduction. They can only asexually reproduce. So that genetic variability comes through the swapping of these plasmids. Okay, let's talk about this process of binary fission. This is how prokaryotes are going to undergo asexual reproduction, which is their only flavor of reproduction that they can do. So the first thing that we need to do is replicate the chromosome. So we're going to make copies of that DNA, and they're going to stay attached to each other, these two chromosomes. And the cell, instead of being nice and round, is going to start to flatten out. Okay. And we're also seeing in the second step, we're seeing these proteins accumulate toward that center of that cell. And then they're going to form this nice little ring, which is going to pull that plasma membrane toward each other until eventually it's going to pinch off. And then we're going to see this this binary fission, right? The separation of these two cells. Each of the daughter cells is going to have a copy of the chromosomes in there. And then once they're all pinched off, then those special proteins are going to go ahead and disperse. Okay, so again, prokaryotes can only do asexual reproduction. So they're going to then, instead of having sexual reproduction to get this genetic variability that we see in eukaryotes, they're going to use this other mechanism called horizontal gene transfer. And this is where we're going to take those plasmids and we're going to swap them between cells. There's three major mechanisms where we can have transformation, where there's pieces of DNA just in the environment, usually after a donor cell dies. They'll just kind of spread all their cell contents. And then some of these molecules might be picked up by the recipient cell. That's called transformation. We can also see conjugation, where these bacterial cells will fuse with each other for temporarily, and the donor cell will provide a plasmid for that recipient cell. We also see transduction, where we can have a virus affect a bacterium, and what the bacterium does is it convinces the bacterial cell's genome to make proteins, and sometimes that one of those viruses, when they go on to spread to the next individual bacterium, will accidentally pick up some of the bacterial DNA instead of the viral DNA, and then that's how that can be induced. Okay, so all of these three, transformation, conjunction, and transduction, are ways that we can provide genetic variability in asexually producing bacteria cells.